Ryan, and I'm going to talk about molecular biology. So part 1, the central dogma. So the overview of the central dogma is a process where the DNA is converted into a functional product, usually a protein, actually always a protein. And in more detail, the central dogma is the flow of genetic information from the DNA to RNA to make the functional product in the protein. And the central dogma was first came up with in 1958 by Francis Crick. Francis Crick also discovered DNA structure. So here's a diagram for the central dogma. So the central dogma from DNA to RNA to protein. So firstly, the DNA would replicate itself, and then it will go through the process of transcription to the RNA. Then it would translate the message to make that protein. So part 2, gene expression. So gene expression, the process of gene expression is the process where information encoded in a gene is used to direct and assemble a protein, which in this case is the functional product. The process of gene expression is used by all known organisms, including multi- and unicellular organisms. It is also used by viruses to make more viruses. And next, transcription and translation in more detail. Transcription is the process where the messenger RNA, the mRNA, is produced to go to the translation happens after the mRNA carries the transcribed message to the ribosomes, and then the message is translated to direct the assemblage of the functional product, again the protein. So part 3 and the last part, the tools of synthetic biology. So the main tool, the only one I'm going to talk about, is genome engineering or genome editing, which is the most basic tool, is where DNA is deleted or modified and replaced into a genome of a living organism like bacteria or a caterpillar. And transcription and translation is already mentioned, so I just won't get into it, and I won't mention the others as well. Others are cellular computation, biomaterials, and regulators, and I won't talk about them much thank you.